Hello, and welcome back to Mammalian Anatomy at Keuka College. We are going to continue our conversation about Chapter 12, the fundamentals of the nervous system and the nervous tissue. Currently, we're still in Part 2, and we are focusing on the nervous tissue. This will be our last video in Part 2. Here we will focus on neuroglia and the role that it plays in creating the myelin sheath. In the last video, we talked about oligodendrocytes, which are found in the central nervous system, and Schwann cells, which are found in the peripheral nervous system. Both of these cells are going to be responsible for making the myelin sheath for its respective location. And the myelin sheath, as we kind of alluded to before, plays a hugely important role in speeding up the electrical current that is going through the neuron or the axon of the neuron. And let's talk about that in a little bit more detail in this video. So, what is myelin? Essentially, myelin is going to be the glial cell, either the oligodendrocyte or the Schwann cell, depending on where we are. And this glial cell is going to take its plasma membrane and wrap it around the axon in kind of a concentric pattern. So it's going to roll itself around the axon. It is important to know that the plasma membrane is lipid-based, and because it is lipid or fatty, it creates a really, really good insulator. So some of the functions of this myelin sheath is, one, most important, insulation. It's going to prevent the leakage of electrical current from the axon. So if one axon in location A is firing, it will not affect an axon in location B. So there won't be any crossover of electrical current from a biological wire to another biological wire. Another function, and equally as important, is that it will actually increase action potential conduction. So it will allow the action potential to move faster across the plasma membrane of the neuron, of the axon. And so it's going to make the signals travel faster. And we'll talk about how it does that in a couple slides. It also helps lower the amount of energy necessary for the action potential to propagate across the axon. So it makes everything nice and energy efficient. So let's talk about myelin. First, we're going to look at myelin specifically in the peripheral nervous system. So we're going to be talking about the Schwann cell. And then we're going to look at myelin in the central nervous system, where we'll be specifically talking about the oligodendrocytes. Okay, so the myelin sheath in the peripheral nervous system. So I want to direct you to this image over here. And this image really shows how the Schwann cell is going to wrap around the axon of a neuron. So in yellow over here, what we have is we have the axon of a neuron, and in blue, we're going to have the Schwann cell, that specialized neuroglia. And when the myelin sheath is forming, what will happen is the Schwann cell will take its plasma membrane and start wrapping it around the axon. And it will wrap around in it'll continue kind of wrapping around in kind of concentric rings, almost like rolling a tube inside of a carpet or something like that. And when it does this, a lot of the cytoplasm and organelles will actually get squeezed out. And so we'll just be left with kind of concentric rings of plasma membrane surrounding the axon. This will form the myelin sheath. This will form the myelin sheath, and this creates a really good insulator within this area. So really, there is not really what's going to happen here is there's not going to be a change in electrical current across the surface of the axon and into its environment here. So, this will act as nice insulation, so electrical current can't leak out over here. It's important to know that when multiple Schwann cells wrap around an axon, they're not going to be butting up to each other or like right next to each other. They're not going to touch. Rather, there's going to be a small gap in between. This gap is called the Myelin Sheath gap. This is a relatively new name because anatomy is slowly moving away from epitomes, like memorial names. I learned it as nodes of Ranvier. So he discovered it and did research on these separate nodes. So there'll be a small little gap between Schwann cell to Schwann cell. And we'll see that the action potential will actually be able to jump over the Schwann cell and go from node of Ranvier to node of Ranvier. This kind of jumping action is called saltatory conduction. So let's talk about saltatory conduction in a little bit more detail. So saltatory conduction actually comes from a Latin word meaning to vault or to leap. And so what happens when there is myelin sheath wrapping around a axon in the peripheral nervous system? Let's talk about it. Essentially, what will happen is rather than having an action potential occurring at each and every single location along the length of an axon, the action potential will actually jump from node of Ranvier to node of Ranvier, or the myelin sheath gap, and it will leap across these areas. Because the action potential is able to leap due to the presence of the myelin sheath, the action potential will be able to propagate and move much faster down the length of the axon than it would be able to do otherwise. I kind of like to think of a ballerina when I think of saltatory conduction. Think of a ballerina leaping along the length of the axon from one point to another. So it can just traverse that distance much, much faster. So if the myelin sheath is present, the action potential can jump or leap using saltatory conduction. And between the Schwann cells, that was a mouthful, and this will speed up the conduction. I think it is also important to talk about cells in the peripheral nervous system that are not myelinated. The axons that are not myelinated are still going to be wrapped with Schwann cells, but the Schwann cell is not going to wrap around the axons in a concentric pattern. They'll kind of be, the axons will be partially enclosed by the Schwann cell. And so what you can see here is we have multiple axons located here, and there can be up to like 15 or more axons within this space. This is just showing a few of them. And what the Schwann cell will do in non-myelinated axons is the Schwann cell will partially wrap itself around each of these axons, kind of grouping them together. But it's not going to wrap around them in concentric rings. It's just going to kind of grasp them and hold them in place. So we want to say that even though these axons are wrapped with a Schwann cell, and we would not say that they have a myelin sheath because they don't have those concentric rings. These type of axons that are not myelinated are going to use continuous conduction or continuous propagation for its action potential, and this process is going to be slower as a result. So in continuous propagation, what will happen is the action potential will have to occur at each and every single point along the axon, and because it does this, it's going to be much, much slower. Oops. And so I kind of think of our ballerina example. Imagine a ballerina dancing on her toes. Imagine how long it would take for the ballerina to move on her toes from one part of the axon to the other part of the axon. It's going to be much, much slower than if the ballerina had the ability to leap. So non-myelinated axons are still going to have Schwann cells, but the Schwann cells are not going to form concentric rings, so therefore non-myelinated axons are not going to have myelin, and it will have a slower propagation of the action potential. So that is our discussion of myelinated axons and non-myelinated axons in the peripheral nervous system using Schwann cells. In the central nervous system, we're going to use oligodendrocytes to form the myelin sheath. It's going to have a similar pattern in which the oligodendrocyte is going to take its plasma membrane and wrap the axon with its plasma membrane in concentric rings, and that fatty layer is going to act as a form of insulation. What is different than that of the Schwann cell is that the oligodendrocyte actually is shaped almost like an octopus. It has many different branches coming out, and it can wrap around multiple axons simultaneously. So not only forming the myelin sheath, it can also function to hold these axons and organize these axons together. Again, like in the peripheral nervous system, there are going to be these myelin sheath gaps that are present. So I call them nodes of Ranvier, but you can use either term. Both are correct. It's just a difference in how we're classifying things now in anatomy. But if we compare these nodes of Ranvier between the central nervous system and the peripheral nervous system, the nodes of Ranvier in the central nervous system are going to be a little bit longer. So just so you know. And because it still has this kind of pattern, we're still going to use, we're going to use that saltatory conduction, that leaping conduction, to speed up the propagation of the action potential. In your textbook, there is a clinical study of multiple sclerosis. Multiple sclerosis is actually an autoimmune disease that will attack the oligodendrocytes in the myelin sheath in the central nervous system. And as a result of that, the action potential can't travel as quickly as it normally would if the myelin sheath was present. This will lead to weaknesses in muscle, possibly blindness, and other symptoms like inability to control urination and those sorts of things. In addition to having the myelin sheath be damaged, the axons themselves will experience inflammation that can kind of worsen these symptoms. Multiple sclerosis actually comes in phases. It will go through phases of I'm forgetting the vocabulary. 1 minute, please. So it'll come in phases of remission and relapse. So it will occur and it'll get better, it will occur and get better. But during the second decade of the disease, it's much more likely that the symptoms will become progressively worse. There are anti-inflammatory medications that can be given to these patients and immunosuppressants that can help with the symptoms of this disorder. So if you want to learn more, go to that section in your textbook. It starts on page 367, and you'll be able to learn about it more in that section. Lastly, I would like to talk about non-myelinated axons in the central nervous system. These may not necessarily be surrounded by oligodendrocytes. They can be surrounded by other glial cells that will allow for there to be insulation present and allow for scaffolding. A lot of these cells are going to be surrounded by astrocytes because astrocytes are the most common neuroglia that are found in the central nervous system, and they serve a lot of different functions. So non-myelinated axons in the CNS or in the central nervous system are still going to be wrapped with glial cells. It just may not be oligodendrocytes. So that comes to the end of our lecture on neuroglia and the myelin sheath. For this section, what I'd like you to be able to do is compare and contrast how oligodendrocytes and Schwann cells form myelin in the central nervous system and the peripheral nervous system, and be able to describe neurons that are non-myelinated in the central nervous system and the peripheral nervous system. Like, how do they differ? What cells are surrounding them? And then be able to compare and contrast saltatory conduction and continuous conduction and tell me which one is faster. Thank you so much for listening today, and I hope you have a wonderful day. Be good to yourself and have fun studying anatomy. Bye-bye. Take care.